My name is Thanawat. Today I will present the quantitative real-time polymerase chain reaction, or RT-PCR, and GATA2 mono allele expression in inherited GATA2-mutated MDS/AML. The PCR is the method to increase the targeted DNA sequences. This technique is very important in terms of gene detection and expression. PCR can be divided into two types. First is conventional PCR, which is the qualitative measurement based on gel electrophoresis. The second one is real-time PCR, which is the quantitative measurements of DNA or gene in absolute or relative values. Today I will focus on the real-time PCR technique. For the background of PCR, Professor Harry Burris invented PCR in 1985, and he subsequently received Nobel Prize in Chemistry in 1993. The PCR consists of the process of applying heating with separate double-strand DNA and adding DNA building block and DNA polymerase that result to form a new chain of DNA. PCR reaction requires three phases of temperature adjustment. The first phase is denaturation. Temperature is about 95 degrees Celsius. The double-strand DNA will be separated into single strand. The next step is annealing, lowering temperature to 68 degrees Celsius to make the primer attach to the DNA strand. And the final step is an extension, heating up to 32 degrees Celsius to allow the optimal reaction of DNA polymerase to occur. This step results in replication of complementary strands of DNA. All of three phases of PCR are counted to each of PCR cycle, and the PCR product will increase in the number in 2 power number of PCR cycles that represent in n or 2 power n of the PCR product. The total PCR cycle is about 30 cycles up to experiment and resulting in billions of PCR production. Focusing on real-time PCR methods, first thing is to prepare the PCR mixture. The target DNA or DNA template along with primer, deoxynucleotide diphosphate as the building block, tag DNA polymerase, PCR buffer, and magnesium chloride will be the ingredients for PCR mixture. The fluorescent signal emission in real-time PCR, along with the increasing of PCR beacons, is the key difference from conventional PCR. The fluorescent signal can be generated by two main techniques. The first technique is fluorescent dye or cyber green, and the second technique is fluorescent probe. One of these fluorescent signal creators will be chosen and filled in the PCR mixture here. The PCR tube will be loaded in the PCR machine, which operates the thermal cycling and computer analyzing that determine the fluorescent signal during the PCR production. For the fluorescent dye or cyber green, this is an agent that can bind to any of the double-strand DNA, and the fluorescent signal will release after its binding. This method is easy to use and quite relatively inexpensive. However, the cyber green can bind in the both specific and non-specific PCR product. For fluorescent probe technique, the probe is an oligonucleotide with one end of fluorescent reporter and another end of quencher. The quencher can inhibit the signal from the fluorescent reporter when the probe is still intact. But when PCR reaction happens at the extension phase, the polymerase enzyme with intrinsic exonuclease activity can cleave the fluorescent reporter away, and quencher cannot inhibit the fluorescent signal. The signal will be released. This method is highly specific to target DNA. However, it takes time to design probe and quite costly. The result from the fluorescent signal during the increasement of the PCR production will be analyzed as the amplification curve, which is the green line in the S-shape here. The curve is plotted between the number of PCR cycles and the fluorescent change intensity. Let's start from the ground phase and increase the slope to exponential and log linear phase, and end with the plateau phase that the curve again to be flattened. The point to be concerned is the second threshold or CT, which is the number of PCR seconds when the amplification curve across the threshold line. There are two main kinds to use the CT value to interpret the amount of gene expression. The first is absolute quantification. This method calculates absolute number of copies based on the standard curve of resolution with none of DNA concentration. The second method is the relative quantification that determines the force change of gene expression between samples and require the reference or housekeeping gene to normalize the value. There's a way to report the expression value from relative measurement. One of the most common is comparative CT or delta CT method. For the last section of my poster, the real-time PCR was used to show the GATA2 gene expression in this publication. The study reports the family member who carry germline GATA2 threonine to 5' methionine mutation. Overall, the 5 generations of family have been explored. The family members who have been labeled in black were symptomatic with MDS or AML or abnormal hematological parameter at presentation. And for the gray shaded were asymptomatic patient or normal blood count at presentation, whereas the white label represent no mutation or no data on GATA2 status. Two family members has been investigated in GATA2 expression gene. Asymptomatic patient number 37 had a single time point, and symptomatic patient number 410 had 4 time point as per third, fourth, 6-year follow-up. The researcher extract RNA from the bone marrow sample and convert to cDNA before using real-time PCR to determine the GATA2 expression, that's shown in this bar graph here. The fluorescent dye was used in the real-time PCR with a relative quantification method to report the level of gene expression. The GATA2 is target gene, and GATA is a reference housekeeping gene. The normal healthy control bone marrow also used with the calculation of the delta CT method. The level of GATA2 expression were higher in asymptomatic carrier compared to symptomatic carrier. Interestingly, the symptomatic patient demonstrated the increase of GATA2 expression during the later follow-up at the 4th and 6th year. The study also provides clinical data with the single sequencing in symptomatic patients. At 1st and 3rd follow-up year, patients with monocytopenia and neutropenia single sequence show mono expression that resulted correlate to cytopenia. And for 4th and 6th year, the blood count spontaneously recovered without the treatment, and single sequence demonstrate the bioexpression. Thank you for your attention.